Hello children, today we are discussing about some experiments with yeast cells. Page number 37, Unit 2, Respiration. So in our book, in 10th class, there are 10 units are there. Nutrition, respiration, transportation, excretion, coordination, reproduction, coordination life process, heredity, our environmental concern. And now we are discussing the lab activity, some experiments with yeast cells. Page number 37. So we need to follow some steps while writing, while doing lab activity. So they are aim, material required, precautions, procedure, observation, conclusion. Here you see figure number 12. There is a diagram, and that diagram is telling you how to do this lab activity. We need to take what? A glass bottle and two hole rubber car and thermometer, delivery tube, and a test tube which is having indicator solution, that is lime water. And now I am telling you what are the liquids to do this activity, and also organisms. We need what? Glucose solution and liquid paraffin, diagen green B, and indicator solution is lime water. Now I am telling you how to do this activity, the procedure. First we need to take glucose solution. So here yeast cells are the organism we are taking for to do this activity. The aim of the activity is to know organisms which can survive in the absence of oxygen or not, organisms which can perform respiration in the absence of oxygen or not. Yeast cells, wild yeast cells, they grow on the skins of apples and grapes, and they derive their energy. Glucose is the resource, a source where it is useful for to perform respiration for the yeast cells. They perform respiration more in the glucose solution. And now we are taking glucose solution in a bottle. We need to heat the glucose solution for about 1 minute. We are heating glucose solution to remove the oxygen, that is dissolved oxygen from the glucose solution. For that we are heating for 1 minute, and later we are keeping it, the bottle, undisturbed without shaking, and we need to cool it. After cooling, we need to add what? Diagen green B or JAYA green B solution in the glucose solution. The diagen green B is blue dye, and it is a reagent, and this reagent we are using to remove if at all there is oxygen present in the glucose solution or not. If we add the diagen green B solution to the glucose solution, if it is turned into pink color, that tells that little amount of oxygen is there in the glucose solution, and it is removed by the adding of diagen green B. So in this way we are making the glucose solution free from the oxygen. Now we are adding yeast cells to the glucose solution. After that we need to add what? Liquid paraffin or wax on the glucose solution. Why we are adding liquid paraffin or wax on the glucose solution is liquid paraffin acts as a barrier between the air that is present in the bottle and also glucose solution. If we don't add liquid paraffin, so here glass bottle consists of air, and air also consists of oxygen, it may dissolve in the glucose solution. And here we are not giving any chance to dissolve the oxygen that is present in the air and the glucose bottle to get mixed with the glucose solution. For that alone we are taking what? Liquid paraffin, and it acts as a layer, it acts as a barrier. It does not allow the diffusion of oxygen into the glucose solution. Later we need to fix that two hole rubber carpet, and we need to insert what? Thermometer, and we need to take the record of the thermometer, that is the liquid, that is mercury level in the thermometer, Again we need to insert what delivery tube, and delivery tube, one end of the delivery tube is in the glucose solution and another end is in the test tube which is having lime water. Here this is the glass bottle, and this is glucose solution, and this is the liquid paraffin, and in this glucose solution yeast cells are there, and we are inserting what thermometer, taking a mercury level reading, and later we are inserting what a delivery tube, and one end is in the bottle and another end is in the test tube which is having lime water. This is the indicator, and after doing all this setup we need to keep it for some time. After that we observed that the mercury level in the thermometer slowly increased, and these yeast cells, they are performing respiration and getting energy, and they are releasing what heat energy and also carbon dioxide, and this heat is useful to heat the mercury level in thermometer, and slowly mercury level in thermometer increases, and they are also releasing carbon dioxide, and this carbon dioxide slowly move and pass through the delivery tube and enter into the lime water, and lime water is turned into milky white, and that tells that this gas is nothing but carbon dioxide, and carbon dioxide is having the ability to convert lime water into milky white. So this activity we are performing to know whether the organisms they perform respiration in the absence of oxygen or not, and here and this activity proves that yeast cell they can perform respiration in the absence of oxygen, and they are performing anaerobic respiration and release heat and also carbon dioxide. And this is a brief description about lab activity, that is some experiment with yeast cell. Thank you.